Hello world. I'm Imagination. Today I'm going to talk about genome editing. Genome editing, or genome engineering, or gene editing, is a type of genetic engineering in which DNA is inserted, deleted, modified, or replaced in the genome of a living organism. Unlike early genetic engineering techniques that randomly insert genetic material into a host genome, genome editing targets the insertions to site-specific locations. Genome editors were available as of 2017. In 2018, the common methods for such editing used engineered nucleases, or molecular scissors. These nucleases create site-specific double-strand breaks at desired locations in the genome. The induced double-strand breaks are repaired through nonhomologous end joining or homologous recombination, resulting in targeted mutations edits. In May 2019, lawyers in China reported, in light of the purported creation by Chinese scientist He Jiankui of the first gene-edited humans, the drafting of regulations that anyone manipulating the human genome by gene editing techniques, like CRISPR, would be held responsible for any related adverse consequences. A cautionary perspective on the possible blind spots and risks of CRISPR and related biotechnologies has been recently discussed, focusing on the stochastic nature of cellular control processes. In February 2020, a U.S. trial safely showed CRISPR gene editing on 3 cancer patients. Genetic engineering as a method of introducing new genetic elements into organisms has been around since the 1970s. One drawback of this technology has been the random nature with which the DNA is inserted into the host's genome. This can impair or alter other genes within the organism. Methods were sought which targeted the inserted genes to specific sites within an organism genome. As well as reducing off-target effects, it also enabled the editing of specific sequences within a genome. This could be used for research purposes, by targeting mutations to specific genes, and in gene therapy. By inserting a functional gene into an organism and targeting it to replace the defective one, it could be possible to cure certain genetic diseases. Researchers have used CRISPR-Cas9 gene drives to modify genes associated with sterility in A.Gambi, the vector for malaria. This technique has further implications in eradicating other vector-borne diseases such as yellow fever, dengue, and Zika. The CRISPR-Cas9 system can be programmed to modulate the population of any bacterial species by targeting clinical genotypes or epidemiological isolates. It can selectively enable the beneficial bacterial species over the harmful ones by eliminating pathogens, which gives it an advantage over broad-spectrum antibiotics. In January 2019, scientists in China reported the creation of 5 identical cloned gene-edited monkeys, using the same cloning technique that was used with Zhong and Hua, the first ever cloned monkeys, and Dolly the sheep, and the same gene-editing CRISPR-Cas9 technique allegedly used by He Jiankui in creating the first ever gene-modified human babies Lulu and Nana. The monkey clones were made in order to study several medical diseases. In the near future, the new CRISPR system will also be able to eradicate diseases and conditions that humans are predisposed to. With this new technology, scientists will be able to take the genes of a human sperm cell and egg and replace the genes that activate cancer or other abnormal or unwanted defects. This will take the stress off from parents worrying about having a child and not able to live a normal life. After just one generation of this process, the entire future of the human race would never have to worry about the problems of deformities or predisposing conditions. Many transhumanists see genome editing as a potential tool for human enhancement. Australian biologist and professor of genetics David Andrew Sinclair notes that the new technologies with genome editing will allow it to be used on individuals to have healthier children, designer babies. According to a September 2016 report by the Nuffield Council on Bioethics, in the future it may be possible to enhance people with genes from other organisms or wholly synthetic genes to, for example, improve night vision and sense of smell. The American National Academy of Sciences and National Academy of Medicine issued a report in February 2017 giving qualified support to human genome editing. They recommended that clinical trials for genome editing might one day be permitted once answers have been found to safety and efficiency problems, but only for serious conditions under stringent oversight. Genome editing tools such as CRISPR are beginning to reshape the physical world around us, one base pair at a time. That's all for today. Thanks for watching.